All right. So, welcome to chapter two. And first, let me apologize again for the mishap of Friday uh, with all the confusion, me being ill and the TA not showing up. I do apologize for the inconvenience. Today, of course, is your online day, so I am giving you a lecture online. Um, it's going to be short and sweet, but it's going to start you off with chapter two. We're probably going to get through the first couple of modules, and then I'll pick up again. Hopefully on Wednesday, I'll be hopefully back up and at it. I finally got my voice back, so um, I'm not going to use too much of it. I'll give you a disclaimer. I'm using AI to help me with this lecture because I am just getting my voice back. So I don't want to strain it. So I will be using AI to help me along the way. And you're going to know the difference. You're going to hear the robotic sounds. It'll sound like me, but it won't be me. And you can tell the difference. So I apologize in advance. But be that as it may, this is going to be about biological foundations of behavior. We're going to go over looking at the functions of the central nervous system as well as the divisions of the peripheral nervous system. Hopefully, you'll be able to learn about the differences within functioning for the sympathetic and parasympathetic divisions in terms of the autonomic nervous system. I'll be able to share with you how the brain will be organized in terms of the roles that the brain plays in terms of our behaviors. And then we're going to look at the different methods that's used to be able to study the brain. As we continue on with the chapter, we'll talk about the two halves of the brain, looking at the different hemispheres and show their different functions. We'll talk about the endocrine system and how it's organized, as well as the roles that our hormones will play in behaviors. Then I'm going to talk about the way we evaluate genetics with behavior, and then also talk about how we as psychologists, we take on studying the roles of genes as well as our environment and how they interact with one another in terms of our behaviors. So with that, we're going to get started on module 2.1, the body's wiring.
Now, I'm sure this is a refresher for most of you, but let's take a little journey through the life of a neuron. Imagine you're at a party. Don't worry, there's pizza. And your job is to pass along juicy bits of gossip. That's pretty much what neurons do, except the gossip is information, and they do it using electricity and chemicals. Our starting point is the dendrites. Think of dendrites as the neuron's many little ears or tree branches. They're sitting there eagerly waiting to hear the gossip, aka signals from neighboring neurons. Dendrites pick up incoming messages and send them toward the main hub of the neuron. Now, the next stop would be the cell body or the soma. The soma is like the neuron's main office. It has the nucleus, the boss, and handles all the cell's day-to-day operations. It also decides, is this gossip juicy enough to pass along? If the signals collected by the dendrites are strong enough, the soma says, all right, let's spread the word. The long road, the axon. Once the soma gives the green light, the signal travels down the axon. Picture the axon as a long hallway or even a telephone wire. Some axons are covered in a fatty coating called the myelin sheath, like insulation on a wire, which makes the signal travel much faster. Without it, the message might just crawl along, and no one likes slow gossip. Time to look at the pit stops, the nodes of Ranvier. Along the axon are tiny gaps in the myelin sheath called nodes of Ranvier. These act like turbo boosters, letting the signal jump down the axon more quickly. It's the neuron's way of using express shipping instead of snail mail. The grand finale terminal buttons, also known as axon terminals. At the end of the axon are the terminal buttons. This is where the neuron sends out its gossip to the next crowd, but instead of just yelling it across the room, it releases chemical messengers called neurotransmitters into the synapse, the tiny space between neurons. The next neurons, dendrites, catch those neurotransmitters and the cycle starts all over again. Boom. Gossip successfully delivered. Here's an analogy to help put it all together. Dendrites listen, soma decides, axon carries, and the terminal buttons share. And that's the neuron's life in a nutshell. It's like the ultimate party network, except instead of drama, it's passing along the signals that let you think, move, feel, and yes, even decide which pizza topping you like best.
You know how we use the neuron, and yeah, I thought the gossip analogy would be helpful. However, just remember, with the neurons, there's three types, okay? We have sensory neurons, we have motor neurons, and then there's interneurons. When we're looking at sensory neurons, which we also call that afferent neurons, here we're looking at how it transmits information from our sensory organs, that being our muscles and inner organs, and it communicates with our spinal cord as well as our brain. Our motor neurons will transmit the information from the central nervous system into our muscles as well as our glands. And then those interneurons, here we're looking at how the information is going to be transmitted from neuron to neuron. Okay? These are going to be the most common type of neurons within your nervous system. The most common type of neuron in the nervous system. Remember that. Okay? Now, the glial cells, here we're looking at how it helps with our nervous system and it supports the neurons, particularly when we're having to get rid of some waste products. And it also provides, like the myelin sheath, insulation to the axon, assisting the neurons in communicating with other neurons. As I mentioned before, the myelin sheath is that layer of protective insulation which covers the axons as well as certain neurons to help speed up the transmission as well as to help with the nerve impulses. I told you before about the nodes of Ranvier, and those are those tiny little gaps that's in between the myelin sheath that kind of boost everything. It's got that little gap to help push together and help move the information forward. One thing I do want to mention though is when we're talking about multiple sclerosis, this is one of those diseases within the central nervous system where the myelin sheath pretty much is damaged or has been destroyed. Therefore, that communication is getting blocked or cut, making it more difficult for our muscles to react the way it's supposed to. Right, we got this time for knowledge check activity. And the question here is which part of the axon would receive messages from neighboring neurons? Would it be the soma? Would it be dendrites? Would it be terminal buttons? Or would it be axon? Do you remember what I said? Okay, so if you thought dendrites, yay, you got it. Dendrites, again, are those branching structures that look like little tree branches that's going to project out from the soma, and it takes in those messages from neighboring neurons. Remember, the soma is the cell body, and that's going to contain the nucleus. The axon, again, is that long cable look that will extend from the soma and carry information away from the soma toward other neurons. That terminal buttons are those swellings at the tip of the axons, and that's going to be where our neurotransmitters are going to be released into our synapses. So just remember, Why the dendrites are important? Because it's taking in and receiving messages from our neighboring neurons.
Okay, we started learning about how neurons chat with one another, looking at the structure of a neuron. Now it's time to discuss what is a resting potential, depolarization, and action potentials. Imagine your neurons as tiny social butterflies that love gossiping with each other. But instead of whispering over lattes, they send electrical signals. Here's how the conversation works. First, we have the resting potential, the neuron's chill mode. When a neuron isn't talking, it's chilling at what's called the resting potential. Potential inside, the neuron is more negatively charged compared to the outside. This happens because of ions, tiny charged particles like sodium and potassium, and special ion pumps keep sodium mostly outside and potassium mostly inside, maintaining this charge difference. It's like the neuron is holding back, waiting for something exciting to happen. Next is depolarization. Here is where the excitement begins. Now imagine the neuron hears some juicy gossip, aka a signal from another neuron. If it's exciting enough, epolar channels open up and sodium ions rush inside the neuron. Suddenly, the inside of the neuron becomes less negative and even briefly positive. Tile's change is called depolarization. It's like the neuron going, wait, what? Tell me more. Last but not least is the action potential. This is considered the big message. If the depolarization reaches a certain threshold, like hitting the neuron's hype limit, it triggers an action potential. It clicks a rapid electrical signal that zips down the neuron's axon like a wave at a football game, floods in, and then potassium exits to reset the charge as the signal moves along. Nickness allows the message to travel fast and reach the terminal buttons at the end of the neuron. So, returning to the analogy about gossip, we now look after the gossip which is resetting. After the action potential passes, the neuron returns to its resting state, back to chill mode, so it's ready for the next message. Potassium and sodium pumps work like the neuron's cleanup crew to restore the resting potential. Time for the recap. Resting potential. Neuron is ready but quiet, negative inside. Depolarization. Sodium rushes in, neuron gets excited. Epolar potential. The neuron shouts the message down the axon. Then it resets and waits for the next message. Neurons communicate like this trillions of times per day, basically making your thoughts, feelings, and late night pizza cravings possible. This is how the action potential works. Starting with sodium, you then see how potassium works along with it. Use chemical messengers to talk. Picture neurons as texters. They don't use words or emojis. They use neurotransmitters to send their messages. Here's how it works. First, the action potential arrives. A neuron sends an electrical signal, action potential, zipping down its axon like a message traveling through a wire. When it reaches the end, at the axon's terminal buttons, the neuron can't just hand off the electricity directly to the next neuron. There's a tiny gap called the synaptic cleft or synaptic gap between them. Then the vesicles will release the neurotransmitters. Waiting at the terminal buttons are little delivery bubbles called vesicles. They're filled with chemical messengers, neurotransmitters. When the action potential arrives, it tells these vesicles, time to send the message. The vesicles fuse with the edge of the neuron and spill the neurotransmitters into the synaptic gap. Third is the crossing of the gap. These neurotransmitters drift across the synaptic gap like tossing paper airplanes across a small space. They travel only a tiny distance, but it's the crucial bridge from one neuron to the next. Finally, you have binding to receptor sites. On the other side, the neighboring neuron has receptor sites waiting like specific shaped locks. The neurotransmitters fit into these receptors like keys, and this binding delivers the message, hey, it's your turn to fire. Depending on the type of neurotransmitter, it can tell the next neuron either to start its own action potential or slow things down. So the question is, why does it matter? Without this chemical handshake, your brain couldn't pass signals between neurons. Every thought, memory, and even your impulse to reach for coffee depends on vesicles releasing neurotransmitters into synaptic gaps and hitting the right receptor sites. So now let's look at this in an everyday scenario. Imagine your friends are trying to coordinate a pizza night. Okay, what can I say? I love pizza. Anyway, this is how it would probably go. Starting with the neurotransmitters, the messages. Your phone sends a text, pizza at 7? This is like a neurotransmitter. The actual message leaving your phone, sending neuron, and traveling to your friends, receiving neurons. The receptor sites, this is who gets the message. Your friends' phones have the right app, the lock, to receive your text. If the app matches the format of your text, it opens it. That's how receptor sites work. Only the right key, message, fits the right lock, receptor. Reuptake, message recall. After you send the text, your phone still has a copy in the sent folder. That's like reuptake. The neuron pulls some neurotransmitters back in to reuse later. No need to keep spamming new messages when you can recycle. So now comes the enzymes. This is the cleanup crew. If someone accidentally sends 10 pizza emojis after the conversation's done, you delete them so the chat isn't clogged. Enzymes do the same. They break down extra neurotransmitters left floating in the synaptic gap so things stay organized. Then the neuromodulators, considered the volume control. Let's say your group chat is on Do Not Disturb for one friend, while another has it pinned and gets special alerts. That's like neuromodulators. They don't send a direct message, but adjust how big of an impact the signals have. Some make neurons more alert, others calm things down. Therefore, your brain is basically one giant super fast group chat where billions of messages fly every second. Without neurotransmitters, receptor sites, reuptake, enzymes, and neuromodulators, all that communication would be chaos, like trying to plan pizza night in a 500-person group text with no rules. How dopamine and certain drugs affect the brain antagonists, the blockers. Some drugs are called antagonists. They block the actions of neurotransmitters by sitting in the receptor sites, like someone taking your seat so you can't sit down. When that happens, the neurotransmitter can't deliver its message to the next neuron. What does dopamine do? Dopamine is one of the brain's most important chemical messengers, neurotransmitters. It's involved in movement, helps you control your muscles and make smooth movements. Learning and memory helps you pick up new skills and store information. Pleasure and well-being makes you feel good when you eat chocolate, listen to your favorite song, or spend time with friends. Social bonding helps you connect with others and feel rewarded in relationships. When dopamine systems don't work properly, If your brain doesn't use dopamine correctly, serious problems can develop. Schizophrenia, a severe mental disorder where people may experience hallucinations, seeing or hearing things that aren't there, have confused thinking, or show unusual emotional reactions. Scientists believe irregular dopamine activity plays a big role in these symptoms. Parkinson's disease, a disease where dopamine-producing neurons are destroyed. Without enough dopamine, people have tremors, shakiness, stiff muscles, trouble walking, and difficulty with fine motor skills like buttoning a shirt. Understanding dopamine and how drugs affect it helps explain why certain mental health conditions happen and how treatments work. For example, know that some antipsychotic medications work as dopamine antagonists to help manage schizophrenia symptoms by blocking excess dopamine activity. Parkinson's treatments often focus on increasing dopamine levels or mimicking its effects to help with movement. We discuss antagonists. So now, what are agonists? Think of agonists as helpers for your brain's chemical messengers, neurotransmitters. They are drugs that either increase the amount of a neurotransmitter available or mimic the neurotransmitter by acting like it and binding to its receptor sites. When agonists are around, it's like turning up the volume on a message between neurons. Examples of agonists in everyday life. Caffeine, mild helper. That morning coffee you love, it's a mild stimulant and an agonist. It increases the availability of glutamate, a neurotransmitter that helps with brain activity and alertness. That's why you feel more awake and focused after coffee. Amphetamines and cocaine, strong helpers. These are potent stimulants and also agonists. They increase dopamine, a neurotransmitter tied to pleasure, movement, and motivation, by blocking its reuptake. When dopamine can't be reuptake, it hangs around longer in the synaptic gap, making its effects stronger. That's why these drugs can cause intense euphoria but also have serious risks. Just remember this key idea. Agonists don't block communication between neurons. That's what antagonists do. Instead, they boost or mimic neurotransmitter activity to strengthen the brain's natural messages.
So we are about to wrap up today's lecture. I'm going to stop here. And this is an assignment that I want you to complete. It will be counted as extra credit of 20 points. See, you're already working on getting extra boost. But also, in fact, it's going to be your attendance for the day as well. So please make sure you answer these just two questions. That's it. You can either use the text entry or you can use Word or Google and upload the file. Either or. But please make sure you complete it by 11:59 today, Monday, August 25th. All right. With that being said, I'm going to say sayonara, and I hope to see you all on Wednesday. Thanks.
Please check your announcements regularly for any changes that may occur. If you have not completed the verification of syllabus enrollment form, please do so immediately so you are not purged from the course. Although I am still under the weather, I will try to communicate with you via the inbox. I hope to see you Wednesday, but if not, I will let you know and I will provide another video lecture. Thank you again for your patience. Hope to see you soon.